Hey guys, so welcome to a lab primer for biotech. This lab is super exciting, and we're finally getting back into lab, which makes me very excited. So next week you guys will be at the bench quite a bit, but what this will do is serve as a bit of an overview or a look at exactly what you're going to be doing and what we're trying to accomplish. Also serve kind of a dual purpose for being an introduction to next week. Right, so we're moving from talking about inheritance and inheritance patterns, which are still a big part of the class because we're talking about genetic diseases. That's kind of our theme, right? But we want to shrink down and scale a little bit. So instead of just looking at an entire individual and what disease they have, we want to start looking at some of the causes. And the great first place to do that is to look at cells. So I have an example here for you. Obviously, you figured out by now that what we're really going to be talking about in class, the model that we'll use in class, is called lactose intolerance. But this is another model of a genetic disease. This is something a little bit different called cystic fibrosis. Right? So what I want to show you over here in this great image, which comes from a company called Vertex, which makes treatments and cures for people with cystic fibrosis, is you can see there's an image of a cell. And in the middle here we have the cell nucleus. And that's actually what they're representing there is human chromosome number 7. And there's a gene on that chromosome highlighted there in yellow that's going to produce something called the CFTR protein, the cystic fibrosis transmembrane conductance regulator. I almost can't even say it. CFTR protein. And those are all embedded here in the membrane of the cell. And in a completely normal version of this, a normal protein, you get a nice even exchange of these little ions here. They're called chlorine ions. In a defective version, remember, and that's usually what goes wrong in a genetic disorder, right? There's a protein that's broken, ultimately caused by a gene on a chromosome, right? So normally what happens here is there's a defective protein, and that doesn't allow these ions to exchange in and out. And when there's no exchange in and out, you get a really poor flow of water into and out of the cells. And that can result in some really sticky mess building up on the outside of the cell here, right? And I have another image that's right over here where you guys can see this takes place in the lungs. Really, so there's the organ, and we're going to zoom in a little bit, and you can see that we're actually in one of the small little tubes here in this branching pattern in the lungs. And then if we zoom in even farther, you can see there's like these little cells which have a really funny shape, with little hairs sticking around on the outside, right? And that's because they're trying to absorb oxygen from blood. And what's going wrong with this disease is they get a really thick mucus buildup because this protein isn't working. And when there's that really thick mucus buildup, you can get bacterial infections, and then people can pass away because of this disease. Right, so this is a little bit closer look with some of the important information. You guys can see this particular image is just a really zoomed up microscope image in which they've stained a few things. And these are the furry things on the outside of the cells that are infected or impacted by cystic fibrosis. You guys see like all the orange there? And I'm trying to point at it in orange. That's the protein. That's the CFTR protein that's being highlighted. And then there's this big blue blob, which is called the nucleus. Right, so those proteins are the things that actually go wrong, and they can cause cystic fibrosis by that buildup of fluid. People get fluid in their lungs that can get infected, and then people pass away usually, in their 30s, really, if they're diagnosed with cystic fibrosis. But ultimately, that's because of a problem with a gene. Right, so again, we're having this connection between phenotype, protein, and gene. Right, so things are going wrong in the cell. Here, you can see these are called histology slides right here, which again is just showing you the cells. This is what somebody would see under a microscope if they cut into some cells, and this would be the normal look of these cells. And here you can see there's somebody with cystic fibrosis with all this mucus buildup and all this mess and what we'd call detritus, which is just garbage floating around. Right, so we can learn quite a bit about genetic disorders, obviously by focusing on the proteins, but also by what's going on at the cell level. There's also some really cool things that we can do with cells, right? We can use them as technology. So this week, the question that we're focusing on is, what is a cell and what do they look like? That's our big guiding question for week number 5. What is a cell and what do they look like? And there's some pretty cool stuff that we can do with that. So as an overview, inside of a cell, you've got all this stuff called organelles, which you learned about in general biology, like a nucleus and the cell membrane. There's macromolecules, which are just big molecules, like proteins, right? Like that CFTR protein that we just talked about. That's one example. There's many, many proteins. Many, 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 many. Hundreds of thousands of proteins. There's nucleic acids like DNA and RNA. There's lipids, which are fats. There's carbohydrates like glucose, right? There's all sorts of that stuff. And then there's other molecules like water and different ions like that chlorine ion that I just showed you, and some oxygen and some carbon dioxide. There's lots and lots and lots of stuff inside of a cell. They're very complex, right? So here what I'm showing you is just a basic overview of cells and what we call their structure and function. Cells are really the basic building blocks of life, right? They're the smallest collection of matter that can be considered living. And all cells start out as sort of blank slates, which is what this image here is showing. And then we're really special organisms because what we can do actually is we can take these cells that are blank slates and we can do a process called differentiation. We can change them into different shapes. Look at all the different shapes of these cells, right? There's a muscle cell which has this long sort of bubble look to it. There are red blood cells, which are really great at carrying around oxygen. There's nerve cells, which look like these really weird branching patterns. I'll show you more of those in a second. There's intestinal cells, which have little hairs on the outside, just like the ones that you saw inside of lung cells, right? And there's a common function between intestinal cells and some of those lung cells. They like to absorb things. In the lungs, that tends to be oxygen, right? But also in intestinal cells, that's food. So they have a really similar shape. But those are very different shapes from things like liver cells and cardiac cells, right? So they can differentiate to perform different tasks. And we need different tasks because we're very, very complex. So there's more types of cells that are going to be needed. And what we want to do this week is we want to look at cells. And then also we're going to manipulate them a little bit and we're going to take something out of them. And that's maybe a bit of a preview for week 6, but that's where we're going. In order to see cells, biologists have to use microscopes. Right? And there's two main types of microscopes that we're going to talk about this week. One is a light microscope. That's what you'll use at the lab bench. And there's also these things called electron microscopes. Man, do I wish we had one because they can take some pretty sweet images. So as an example, here's something that a light microscope can look at. These are little teeny bacteria, right? This is a really cool view of something called E. coli. But if you had a light microscope, look at all this detail you can get to. Right? We'll talk about why this happens, but light microscopes, you can get these really, really super high resolution images. Another example are these things. Can you tell what those are? Because I showed you on a previous slide a cartoon version of these. These are nerve cells. So this is what they look like underneath a light microscope, but look what they look like underneath an electron microscope. And look at all that detail. Right? So by using electron microscopes, we can get a lot more detail because of high resolution. We're going to use light microscopes and look at things like this week, right? We're going to look at these types of patterns. So, as a recap in this lab primer, cells are the fundamental units of living things, and they contain a lot of things inside of them. They're wrapped up in a membrane. They have a lot of proteins on the inside. They're all shaped different with different molecules. We have DNA that are packed up in the inside, and we can see them actually with pieces of technology called microscopes, and that really helps us understand their functions. At the bench this week, you'll see more what cells look like, what their key components are, also how they're organized, and a lot more, right? And we're going to do that through two main techniques. Number one is cell staining. So in order to see them underneath a light microscope, cells aren't normally colored, right? They don't provide us with the ability to see them very easily. So what we're going to have to do is we're going to have to stain them with a special type of stain called methylene blue. We're going to have to also visualize them underneath a microscope. Now, I'll go over with you like some of the basics of a microscope, but to me it's kind of like learning how to use an iPhone, right? You don't necessarily look at the lab manual to figure it out. You just kind of start playing with it and you'll figure it out. But we'll go over that together when you're actually in the bench, like how to use these light microscopes. The other thing I'm going to have you do this week is you're going to have to collect some cells, right, from you as one source. And you're also going to have to stain them. And we're not going to need a ton of liquid to do that. So we're going to start using something called a micropipette. And this gentleman right here, I'm linking the video to this page. I want you to check this out because he's going to give you some micropipette tips. Right, so that's something that we use in biology is we use pipette tips and we use micropipettes. And you'll see that this week. But what I'd like you to do is watch this quick video and he'll give you a really brief introduction of what a micropipette is and how we use them. You'll be using them this week in lab. Both days that you're with me, you'll be in the lab. And we've also got some really cool remote assignments planned for the days that you're not with me. So thank you so much for watching this lab primer, guys. Hopefully this gives you an idea of what we're looking at next week, how we're going to accomplish that, and the main topic being cells.